Good morning, folks. Thursday morning, about 9:45, and some of you may be involved with your exam 4. I hope you're doing well with it. Rest of you, I guess, are studying, working, or something occupying your time. Anyway, we want to finish chapter 12. So we talked about the central nervous system, the brain, and the parts of the brain. And so now what we want to do is look on page 443. 443, and you look in the top, it says module 12.3, protection of the brain. And so you come down to where you see the cranial meninges. Now, as soon as you see meninges, you probably have heard someone talk about meningitis. Serious situation. Number of organisms can cause it, all the way from bacteria to viruses to fungi to protozoa. So it's a serious infection. People can die from that. Some mortality rates are about 85% if you don't do something for it. Don't take any aspirin and just figure you're going to ride it out. They may carry you out in a couple of days. But anyway, as you look at the cranial meninges, you come down to the bottom of page 443 in that first column, and it says they're protective membranes, primarily of dense, irregular connective tissue. Here we go right back to chapter 4, don't we? You see how this stuff builds? And of course, dense, irregular, collagenous connective tissue is pretty strong stuff. So the outermost meninx, that's singular for meninges, the outermost meninx, you see in the bold print there, is the dura mater. And so it's up next to your skull, very tough. If you look at the text below dura mater, you see it says it's the thickest and toughest of the membranes. So there's an epidural space, as you see, above the dura mater, which is that space between the dura mater and the skull. And then you come down to see a subdural space. There is a space that's below it. Now, the way to understand this, I think, is probably to look over on page 444. We'll flip back and forth to some degree on these two pages. So as you look at figure 1218, and we're not going to look at the top, left or right. We're going to look at this frontal section of the brain with the meninges. So as you look at this picture, you see over on the left, you see the skull. Remember intramembranous ossification? See how you have a solid plate of bone, and then you have the trabeculae, spongy bone, and then you have the outer bone there. So you see the skull, and look to your right, and all the way over to your right, and you see a couple of terms, periosteal dura, meningeal dura. We're really just going to call it all one thing. Just going to call it the dura mater. And so that is the outermost layer. Now, it said back on the previous page there was a subdural space. So sub means underneath, like submarine. And so there is a space underneath the dura. Sometimes people have hemorrhages, and they have a subdural hemorrhage. So now you know what they're talking about. Serious things. Need to get it taken care of if we can as soon as possible. So there's the dura mater. You can just put dura mater beside, draw your little bracket beside periosteal dura and meningeal dura, and just call it the dura mater. And then you see the subdural space. And then you come down under subdural space and you see Arachnoid Mater. Excuse me, most of the time they don't include the word mater. They just say the arachnoid membrane. And of course, some of you probably have, I think there was an old movie called arachnoiditis or arachnophobia, something like that. And you may or may not know, but arachnoid refers to spiders. So obviously we don't have spiders running around in our head, but we have a structure that encourages people to think about the webs that spiders make. And so you come down under subdural space and you see the Arachnoid Mater. And if you look closely, you can see the membrane, but you can also see these little lines coming from that. And so if you will look at the right side of the brain, come into the brain, you see arachnoid trabeculae. You remember the trabeculae in spongy bone? Okay. So those are little structures that help support the arachnoid membrane, and they attach to the next layer called the Pia Mater. Now, before we get to the Pia Mater, you see this space called the subarachnoid space. So you see the blood vessels are in that area. You can see the vein there that's blue, and you can see the artery to the left, to the right. To the left, and the vein to the right. So sometimes when people have a stroke or have a hemorrhage, cerebral hemorrhage, that blood is starting to fill in with in that subarachnoid space, and that can create pressure and problems for the tissue. The brain's, not as tough as some things, so you get some pressure on it, you could damage some tissue. So we've got the dura mater, the subdural space, the arachnoid mater, and then we have the subarachnoid space. And we're going to find out that there's fluid in there. We're going to talk about that. You come down to the last one called the Pia Mater, and I think over here it gives you a definition for that. Pia Mater means gentle mother. So it's not a real thick, heavy membrane, but it is right on top of the gyri and the sulci that we talked about, or sulci, however you want to talk about it, however you want to pronounce it. So those are the layers of the meninges. And if you look under arachnoid mater down at the bottom of the page, let's see, where did I see that? Look in the second line. Like the dura, the arachnoid mater is composed of dense, irregular collagenous tissue. However, it's thinner and more elastic. So you got this tough layer outside. You got a similar layer, but it's thinner and therefore not as tough, more elastic. And then you come down to the Pia Mater, and it's what actually touches the brain. Very thin membrane there. So we've got those three membranes. Now I want you to notice a couple other things before we get into the flow, the manufacture, and the flow of cerebrospinal fluid. So we're looking still at this structure, the third structure, the one down toward the bottom. And I want you to move toward the midline, and you see this sort of triangular structure called the superior sagittal sinus. That's right up in the middle. You remember the longitudinal fissure we talked about? You can see how this is all building on what we've already covered back in the early part of chapter 12. It's not a real blood vessel in that sense, but it does collect blood, venous blood, from the brain and brings it back to the heart, eventually to the right side of the heart. And of course, the heart pumps it through the respiratory tract so it can pick up oxygen, and then sends it to the left side of the heart, and the left side of the heart pumps it all over the body. So you want to be familiar with that superior sagittal sinus, and you see this little structure called the arachnoid granulation. Now they used to call them arachnoid villi. You remember the microvilli that we talked about in the columnar epithelium in your small intestine, increase the surface area for absorption. They used to call these things arachnoid villi, but now they call them arachnoid granulations. But notice that the arachnoid granulation is connected to the subarachnoid space. So you can imagine, you might think, well, maybe blood comes from that superior sagittal sinus and goes into the arachnoid villi. Well, it actually happens the other way. We have cerebrospinal fluid, which we're going to talk about in a minute. Our brain and our spinal cord float in that fluid. And we're always making that fluid, and it eventually gets into the arachnoid space, subarachnoid space, and then it goes out of the subarachnoid space into the superior sagittal sinus, which is draining venous blood from the brain. And venous blood is going to go back to the heart. Okay? Now, having said all of that, let's look on page 445. This might be a little bit challenging, but I believe you can handle it, even if you can't quite visualize it. As you look at figure 1219, you see it's labeled the ventricles of the brain. Ventricle is a cavity. And so you look up to the left of that brain, it says anterior horns of lateral ventricles. Now, we have a lateral ventricle in this hemisphere and a lateral ventricle in this hemisphere. If you look closely, there's a little bit of a separation there. You come to the lower part of the cerebrum on that illustration, you see inferior horns of the lateral ventricles. And so you've got two cavities, one cavity in this side and one cavity in this side. One cavity in the left cerebral hemisphere, one cavity in the right cerebral hemisphere. Interesting shape, isn't it? It's not just a void or anything like that, but it's got a shape to it. And you see they name all of those things. Like if you look to the right, you'll see that they will have a label called posterior horns of the lateral ventricles. You don't have to know about it. All you need to know is you have a right lateral ventricle and a left lateral ventricle. Now, what's not showing here, and you can look on page 446 for just a second. As you look on 446, you look up at the top left, you see superior sagittal sinus. We're going to put all this together in just a minute. When you come down on the left, you see the choroid plexuses of the lateral and third ventricles. So they're pointing out, you take the top line, and that's in the lateral ventricle. So the right lateral ventricle, cavity over there, has a collection of capillaries called the choroid plexus. And from that collection of capillaries, we have produced a fluid called the cerebrospinal fluid. It's somewhat like plasma, just to make it somewhat relative to what you have, or relevant to what you already know about blood and plasma. So each lateral ventricle has a collection of capillaries called the choroid plexus, and they produce cerebrospinal fluid. Okay? Now, that fluid's got to go somewhere. It flows. It is not stationary. It is flowing. And there are cells that you already know about that cause that fluid to flow. You have the pressure in there of when you make new fluid, it pushes the older fluid around. But you also have some cells. I'm going to let you look it up. And well, you might find that in chapter 11, when we have this test in exam number 4. So, because we keep adding to it, just taking the foundational material and we're adding to it. So we have these cells. Now I'll let you look that up and refresh your memory about that. But they help move the cerebrospinal fluid produced by the lateral ventricles, choroid plexuses. And if you look back on page 45, 445, you see this little junction. Look at the anterior horns of the lateral ventricles, and you see down below there, you see this little tiny blue, kind of blue-green, is that what you want to call it? I don't know what color that is. But anyway, it's a connection to the third ventricle. And if you look to the right, between these two models, two illustrations, you see interventricular foramen. They used to call it the foramen of McGinty. I think it's McGinty or Monroe or something like that. And you don't know where in the blazes that is. It could be in your nose. It could be in your toenails. We don't know. But they changed it now so that it's really more meaningful. Interventricular foramen. So you got this opening where both the lateral ventricles empty cerebrospinal fluid into the third ventricle. Now that third ventricle kind of looks like a bird's head, doesn't it? So it's a very narrow cavity. Remember, as a matter of fact, look over to the right, look at the anterior view, and look at the bottom right hand of that illustration, and you see diencephalon. Now that should ring a bell like hypothalamus and thalamus. The large structure you see there, that's thalamus. You got a thalamus on either side in either hemisphere. Actually, the walls of the thalamus help make up the walls of, if you look in the middle, you see it says third ventricle. So the walls of the thalamus help make up the walls of the third ventricle. Now, some of you might want to know what that little brown structure is in the middle of that third ventricle. They've got some special name for it, and I've forgotten it right now. You don't have to know it. It's like the interthalamic protuberance or something like that. But you don't have to know that. Anyway, that's it's a small cavity, and as you look to the right and you look at the third ventricle, it's narrow. Now it's got some depth, but anterior to posterior, but laterally it's very narrow. So here we have the third ventricle. Now go back over to page 46, 446, and you see where it says on the left-hand side, choroid plexus is of the lateral and the third ventricle. So there's a choroid plexus in the third ventricle, producing cerebrospinal fluid. Let's go back over to page 445. Now remember, this fluid is flowing through these ventricles. So as you look at that little bird head, don't put that on the test, okay? Just the third ventricle. But you see that little, looks like a skinny little neck. You follow that down, and you hit this structure called the Cerebral Aqueduct. Look in the middle between the two illustrations. Then a little canal, and so that canal guides cerebrospinal fluid into the fourth ventricle. So we have four ventricles: a right lateral ventricle, a left lateral ventricle, a third ventricle, and now you have a fourth ventricle. So as you look on page 446, and you come down to where you see the pons and the medulla and the cerebellum, and look right underneath the cerebellum, and you see this kind of reddish structure like you saw up there in the third ventricle and the lateral ventricles. And you see it says the choroid plexus of the fourth ventricle. So we have four choroid plexuses, collections of capillaries that produce cerebrospinal fluid. Now, if you look back on page 445, the fourth ventricle in the left illustration looks pretty small, doesn't it? But look over on the right. Look at the right illustration. The fourth ventricle is wider than it is tall. Okay? And if you follow the fourth ventricle, that fluid is going to go into two places. One thing it's going to do is you go back to page 445 and look at part A, the lateral view. Look at the last term down at the bottom in the middle. You see central canal of the spinal cord. So fluid flows down the very center of the spinal cord. Now, eventually what's going to happen is that's going to, the cord itself is going to end. We'll talk about that a little bit later. And the fluid comes out and fills up the arachnoid space, the subarachnoid space, in the vertebral column. And so your cord floats in fluid. That's protection. And of course, we get into all kinds of twisting our body this way. So it gives a lot of protection to the cord. Now, I want you to look over on 446 again. And you're looking at the cerebellum and the pons and the choroid plexus of the fourth ventricle. And you see where it goes down the center. But look at the arrow that goes to your right. And you see that arrow goes around the cerebellum, okay? Some of it goes down into the subarachnoid space around the cord. You see some of the arrows coming back up. See, here's this circulation I told you was occurring. You follow those arrows, and you can go all the way around the brain. So our brain floats in fluid. It's like a shock absorber. It protects us. Now, I want you to look at where the I'm on 446. Look where it says choroid plexus of the fourth ventricle. Follow the arrow. You see the fluid goes to the left. Oh, and it goes out. One of those arachnoid granulations. Fluid keeps on going. More fluid goes in through the arachnoid granulation. Look at the top left. The fluid goes into the superior sagittal sinus. So CSF, short for cerebrospinal fluid, comes from blood and goes back to blood. You know, we're getting into recycling right now, right? Yeah, try and reuse things, turn them into other stuff. Well, that's what we're doing with the cerebrospinal fluid. It flows around our brain, so we always have fresh fluid, and around our spinal cord, and it gets back into the bloodstream through the arachnoid granulations, gets into the superior sagittal sinus, and that is not really a blood vessel, like I said, it's a sinus, but it collects blood, and then simply the blood flows back down. You've probably heard of your jugular vein. Well, that's one of the places it comes down on both sides, and eventually gets back into a subclavian vessel, and then that joins with a brachial vessel, axillary vessel, and that goes to eventually the superior vena cava. So it's a big cycle. Okay. But the purpose of that fluid is to give support and protection to our brain and our spinal cord. As you look on page 446, they've got 1, 2, 3, 4 sort of steps about the flow of cerebrospinal fluid. You think I ought to get you to know that? No, Mr. Princey, no. Yeah. So you need that, okay? So make sure you know the flow of cerebrospinal fluid. And then in the second column, I want you to look at the second paragraph. You see the term hydrocephalus, which means water on the head. Sometimes people are born with a defect, or they may have some sort of a event that damages something so that the fluid, the CSF flowing, is obstructed. And then you don't stop making fluid, but it can't get back to the superior sagittal sinus, and the head starts swelling. Now, if you're an adult and you have that happen, your head can't spread because these bones have already cemented themselves. Now, in a baby where those are not solid yet, they have not joined tightly. We call them fontanelles, remember that? And those fontanelles will start to spread apart. And it's giving, so the pressure is pushing those bones out. So you used to have to go in there and they have to put in an artificial drain, and they'll often put it in back here, and it'll come across the chest, and they'll put it under the skin so that it eventually drops into the fluid drops into the abdominal pelvic cavity. It's absorbed and it reduces that pressure. Anyway, you can read about that. Okay? Now, let's move to 448. 448. And we're looking at module 12.4, the spinal cord. As you look in the second column, you see it's a relay station. It relays information from the periphery to the central, and it sends messages back down from the central out here to the peripheral nervous system. And you see it's a processing station. We think of processing is up here, but there are some circuits, a motor neuron, a sensory neuron, and so forth. And then you have a central area, sort of, we would call it a control center. They establish reflexes, which can be very quick. And those reflexes often can be used to indicate a healthy body or a body that's got a little bit of a problem. Most of you have had a physical, and one of the things that the doctor did was have you sit on the edge of the examination table, and he took a little rubber hammer and tapped right below your patella, and that usually makes your leg jump, your lower leg. And so that's a reflex that does not have to go up here in order to occur. It just goes right into the spinal cord and comes right back out, and your leg swings up because you've overstretched the patellar tendon. So that's why that happens. But it's an indication of health, and they do that with newborn babies too, some with their feet and so forth, to see if they have these reflexes. So anyway, that gives you a little bit of an idea of the primary roles of the spinal cord. So let's come down to where it says protection of the spinal cord. Just like the skull floats in fluid, as I've said before, so does your spinal cord float in fluid. So it's there for protection. Now you already know the layers, you already know the membrane. So as you come down into this paragraph, if you come down 4 lines, you see over to the right the 3 meninges, okay? But I want you to come down to line 8, and I want you to go to the middle of line 8, the middle of the paragraph. It says the spinal pia plays a role in cranial, a role that the cranial pia does not. It helps to anchor the spinal cord in the vertebral cavity. How does it do that? It does it through denticulate ligaments. Denticulate ligaments. If you will look on the facing page and look at figure 1222, now you got 3 components to 1222. You got the upper one for the person's head. You got the middle one, which illustrates the membranes that you already know. You want to know those. And then you look down at the cross section. Now notice in the cross section, you see anterior at the bottom and you see posterior at the top. Okay, getting oriented with those terms. So you look on your left and you can see where they have dura mater, arachnoid mater, and pia mater. See the kind of, see what the epidural space is filled with? Got some fat in there. It's good to have some fat. It's a cushion. Yeah. Got some ventral cushion too, don't we? That's all normal. Again, homeostasis, the right amount. You don't want too much, you don't want too little. And so as you look to the right, you can see the epidural space, which is filled with fat, subdural space, subarachnoid, and here's the ligament. Here's where the pia mater of the spinal cord does something that the cranial pia does not do. You see that it has a ligament, and it attaches the pia mater to the spinal dura, called the denticulate ligament. So that helps to stabilize it. Could you break it? Sure you can. Just like people, sometimes I remember a football player for the Atlanta Falcons, strong runner, and he cut really hard and snapped his ligament, anterior cruciate ligament in his knee, just ripped it right apart. So obviously we can exceed the capability of some of our ligaments, but this is meant to hold it somewhat stationary and stable. Now, before we go on to another page, I want you to look to the right on page 449, and you see epidural anesthesia and lumbar punctures. I would like for you to read that, because some of you have maybe had a baby and you had a epidural anesthesia. Okay, epidural. Where does that anesthetic go? Into what space is it placed? And then you can read about the lumbar puncture. And this is where we pull out the cerebrospinal fluid to look at it under the microscope to see what might be causing a problem for a person. Okay, now we are going to go over to page 450. And there's some terms about the spinal cord I want you to know. So as you look in the first column, 450, external spinal cord anatomy, don't worry about the sulcus or anything like that. Come down, conus medullaris. Conus medullaris. Now that you're not careful, that'll fool you. You'll be thinking it's up there in the brain. Okay. But it's a little cone-shaped end of the spinal cord. And then you see the next bold print term says phylum terminale. Okay. And you see what that does. It anchors the spinal cord in the first coccygeal vertebra. Our coccyx, you remember that bone? Fused down there at the bottom, a little tailbone. So that's where that phylum terminale comes in and helps to anchor the cord in another fashion. Look at the second column and you see the bold print terms, the cervical enlargement, the lumbar enlargement. Notice it says the nerve roots that fuse to form the spinal nerves serving the upper and lower limbs attached to these enlargements. So we have a cervical enlargement, we have cervical nerves that come out, and then you have a lumbar enlargement, and that's where the spinal nerves come out from that particular point. The last thing you see is cauda equina. Cauda, caudal, or cauda means end or tail, and equina, and so that means horse. So if you look at the figure 1223 and come down the cord until where you see it says first and second lumbar nerves, and then from then on you don't have, after the conus medullaris, you don't have a solid cord. You have these nerves that run down through the vertebral foramina. And they eventually end down at the bottom, down in the sacral area and so forth. So you want to know those structures. Now let's look on page 451. Internal spinal cord anatomy. We already know there's a central canal because we mentioned that back when we were talking about the flow of cerebrospinal fluid. I want you to look at the illustration down below, 1224. You want to know those structures. You want to know the positions, anterior and posterior. Coming down the left-hand side, gray commissure. Now that whole thing that looks like a butterfly in there that's brown, that's all gray matter, non-myelinate. A commissure joins the two, can we say the wings of the butterfly? They talk about that. And you notice that the wings have parts, labeled parts. So you have a posterior horn. You hear horn, you think gray matter. You have a lateral horn, and you have a ventral horn. Now you can look right above there and you see anterior horn, posterior horn, lateral horn. And of course you can have right posterior, left posterior, and so forth. So that's gray matter, and you want to read the anterior horn, posterior horn, lateral horn. Where, what is their function? Where do they go? Okay, nothing hard there. Just make a little chart and work that out. And then you see over on the right, they show you that the blue up at the top, the posterior horn, and they've got a little enlargement called a posterior root ganglion. Now follow those blue lines and you see they terminate in somatic sensory and visceral sensory receptors. So these blue lines coming in the posterior root, as you see on the illustration, they're sensory. They're picking up signals from our environment, perhaps, or our skin, something like that, or from our organs, our viscera. So they bring messages in, and then messages obviously go out of the spinal cord. If you look down at the lateral horn and the ventral horn, both of those are motor. So you see those red ones going out into that ventral root or anterior root. You have a posterior root with a ganglion up top. Now you don't have a ganglion down here. But the anterior root carries motor neurons out to viscera, as you see they're talking about the lungs, or your skeletal muscles. Now when I do that, that means some information is coming out of the cervical region of my spinal cord, and it comes out the ventral root, horn, and enters the anterior ventral root, and then goes to skeletal muscles. Now, a couple of things here I want you to realize. You see over to the right, not all the way to the right, but look where you see posterior root ganglion. Right below it, you see spinal nerve. So you want to realize that spinal nerves are mixed nerves. They have sensory and motor, or you could say afferent and efferent fibers. Now they'll eventually split as they get farther away from the spinal cord. The sensory will go where it's supposed to go. The motor goes where it's supposed to go. Isn't it amazing that they do that? All in 9 months, constructing the body that we're going to live in. I love this stuff. I love it. Just great. Now, one thing I would like you to know that's not going to be listed here. When you look at the anterior horns, or you can call them the ventral horns, that's where the polio virus attacks. Now, not everybody gets that attack. Some people just get a headache, they get a sore throat and a fever, and that's it. But when somebody's paralyzed, the news media gets on it, and then it gets somewhat blown out of proportion. But anyway, that's where the virus likes to strike, if it gets that deep into our bodies, at the anterior horn, which is motor, so it could destroy to some degree the ability to control a leg, a foot, an ankle, or something like that, or a hand or arm. And that's where you get some paralysis. But you, just like me, took polio shots, and we're okay. Except they're going to want you to get shot again. I think when I was around 50, gosh, that was 23 years ago. I think I was around 50, and the doctor said, I want you to get a Zostavax. That's a vaccine for chickenpox, because the virus that causes chickenpox also causes shingles. Okay. It's in that virus is in our body. We don't get rid of it if you've ever had chickenpox. I had it because we didn't have a chickenpox virus at that time, vaccine at that time. But anyway, polio virus likes to attack that area, and we got the polio vaccine. Page 451, second column. I want you to read down a little bit with me where it says spinal white matter, ascending and descending tracts. Ascending tracts, remember tracts, those are axons, myelinated axons in groups, and those are called tracts in the central nervous system. They're called nerves outside the central nervous system because they start to split up. But they call them tracts, and they can go up. That would be a sensory tract, or if they're coming from the brain down, that would be a motor tract. I want you to look at something on page 452. On page 452. Now, don't go memorize everything that I'm going to show you. I'm going to point out what I want you to know. As you see on page 452, first column, you see ascending tracts. They're sensory. They're afferent. You move over to the right and you see descending tracts. They're motor or efferent. Now, they name these tracts. Many times they name them according to their origin and their termination. Look at the figure 1225. I want you to look in that box. Come down 1, 2, 3, 4. Now notice the name of that tract. Spinophthalmic Trap. Does that register with you? Does it make you think about the origin and determination? The first part of the word is the origin. So this originates in the spinal cord, and it terminates where? In the thalamus. So you've learned a lot of vocabulary, and here we are putting together words that are meaningful. Okay, so that's a sensory tract, right? Or an afferent tract. It's going to carry messages up. Come down to the next box. We're going to look at a descending tract, and I want you to come to the first block It's the big block, and come to the first small block, and it says corticospinal tract. Originates where? The cortex of the cerebrum. Maybe also the cortex of the cerebellum. And it terminates in the spinal cord. So let those names teach you something. You've learned a lot of vocabulary. Apply it, all right? So that would be a motor tract, wouldn't it? Okay? That's all I want you to know about that. I think there was one other thing I wanted to go over. Let's see here. Yeah. Go to page 463. Page 463. And look at module 12.7. Homeostasis. Oh my, we've got to go back to homeostasis. What did I tell you at the beginning of the semester? Keep it in mind. It just keeps coming back up. See, that's foundational. We've got all these homeostatic mechanisms, which most of the time they talk about temperature, talk about your sugar level or your blood pressure. But there are a lot of different homeostatic mechanisms. Come down in that column to the bottom where it says homeostasis of vital functions. And you don't have to think about it. You are engineered so that you keep your body healthy without you having to think about it. You might have to eat the right foods, but let's look at it for a second. The homeostasis of vital functions such as heart pumping, blood pressure, and digestion is controlled by the autonomic nervous system. You and I don't have to think about it at all. Probably in about 45 minutes, I'm going to be hungry. My system's going to say, get something, Mr. Pretzel, get something. Now, having said that, we'll remember that. And then I think it's the last thing I want you to know. Let's see if I don't think I had anything else after this. There's so much here, but you're not going to be a surgeon yet. So let's just get the basics down. Look in the second column on page 463. Second column, second paragraph. The main boss of the ANS, the autonomic nervous system, don't call it the automatic, okay, which receives sensory input from the viscera, the components of the limbic system, which we didn't cover, and the cerebral cortex. We mentioned that. That's where you do your thinking. This allows the hypothalamus, right below the thalamus, to respond to both normal physiological changes and emotional changes, to adjust the autonomic nervous system output, to preserve homeostasis. Look at the bullets down below. Don't go memorize them. Just look at other centers. Many nuclei. Remember, that's a little collection of nerve cell bodies, unmyelinated axons. Many nuclei in the reticular formation participate in regulation of your digestive processes and control of urination. Yeah. All you and I got to do is recognize when the bladder's full and find a place to sit. Get it all done. But it helps to keep it in homeostasis. Okay? All right, that takes care of part 2 of chapter 12. I'll be working on part 13. Well, not part 13, but chapter 13 and chapter 14. So we'll get that done next week. Okay? All right, well, I'm going to end this. That's what I want to do, is go about 45 or 50 minutes. So do well. I hope you did well on your fourth exam. I really like to see you succeed, but I got to put you through some rough stuff, don't I? Yeah. Kind of like going into basic training. Got to teach you how to survive. Okay? So y'all have a good weekend, and Lord willing, we'll see you next Monday.